Good morning, everyone. Today I will take you on a journey inside of the cell to explore one of the most fundamental processes in biology, cellular respiration. We will see how a single molecule of glucose is transformed into the universal energy currency of life, ATP. My talk is structured into three main parts. First, an overview. What is cellular respiration? Second, the core of the process, a detailed look at three-stage pipeline, glycolysis, the citric acid cycle, and oxidative phosphorylation. Finally, we will do the energy accounting and discuss its profound biological significance. So, what is it? Cellular respiration is a metabolic process where cells oxidize organic molecules, primarily glucose, to produce ATP. Its main factory is the mitochondria. This overall equation summarizes the transformation. One glucose plus six oxygens yield six carbon dioxides, six waters, and energy. However, this doesn't happen in one explosive step. The cell uses a brilliant step-by-step strategy to extract energy efficiency. Please look at the central diagram. It's a three-stage pipeline. Glycolysis happens in the cytosol. It's the initial splitting of glucoses. The citric acid cycle takes place in the mitochondrial matrix, where the breakdown continues. Finally, oxidative phosphorylation occurs on the inner mitochondrial membrane, where the bulk of ATP is made. The strategy is to release energy gradually and trap it in small, manageable amounts. And here is a three-stage pipeline for energy extraction. It includes the stage with overview and energy strategy. So let's enter the pipeline stage one, glycolysis. It means sugar splitting occurs in cytosol. Please focus on the content. It's a 10-step pathway that follows energy investment, then payoff logic. So first, two ATP are invested into the phosphorylate and oxidative single glucose molecule. The six-carbon sugar is then split into two three-carbon molecules. In a payoff phase, three molecules are converted into two molecules of pyruvate through substrate-level phosphorylation. Four ATP are produced directly. So the yield, two pyruvate, two net ATP, and two NADH. The pyruvate now travels to the mitochondria. Stage 2 begins as pyruvate enters the mitochondrial matrix. So first, it undergoes a bridge reaction. It is converted to coenzyme A, releasing one carbon dioxide and generating one NADH per pyruvate. Coenzyme A then enters the citric acid cycle, also known as the Krebs cycle. This is the overview and how the energy is produced and the carbon fate. This diagram shows the cyclic nature of the process. It's an 8-step cycle. The core mission here is twofold. Completely dismantle the carbon skeleton and harvest even more electrons. In each turn, the two carbons from coenzyme A are released as two carbon dioxide molecules. More importantly, through multiple oxidation steps, the cycle generates a large haul of electron carriers, 3 NADH and 1 FADH2 per turn. One ATP is also made directly by substrate level phosphorylation. Remember, one glucose gives two pyruvates, so cycle turns twice. The total contribution per glucose is 6 NADH, 2 FADH2, 2 ADP, and 4 carbon dioxide. Now, stage 3, oxidative phosphorylation. This is where the magic happens. On the inner mitochondrial membrane here, NADH and FADH2 cache in their virtues for ADP. This is the most critical diagram. Please follow closely. First, electron transport. NADH and FADH2 donate their high-energy electrons to protein complexes embedded in the membrane. The electron transport chain, ETC. Electron pass from one complex to the next like a bucket brigade. As they move down the energy gradient, they release energy. This energy is used to pump protons from the matrix across the membrane into the inner membrane space. This creates a high concentration of protons outside, like water behind a dam. The result is an electrochemical gradient rich with potential energy. Oxygen is vital here. It weighs the end of the chain as the final electron acceptor, combining with electrons and protons to form water. This removes used electrons and keeps the chain flowing. Now, the payoff. Chemiosmosis, stored in the proton gradient, is forced to make ATP. The enzyme ATP synthesis acts like a molecular turbine. Protons flow back down their gradient through ATP synthesis into the matrix. This rush of protons spins a part of the enzyme, driving the mechanical synthesis of ATP from ADP and a organic phosphate. This process, oxidative phosphorylation, is how the vast majority of ATP is made. Each NADH feels the pumping that leaves at least maybe about 2.5 ATP, and each FADH2 about 1.5 ATP. So energy balance. Let's do the accounting here. Adding up all the ATP from three stages, glycolysis gives a net of 2 plus ATP from its NADH. The citric acid cycle gives 2 directly plus ATP from its many carriers. The grand total from one glucose is approximately 30 to 32 ATP. This process converts about 34% of energy is glucose into ATP, a remarkably efficient biological engine. Beyond energy, respiration sustains life and ecosystem. This ADP powers virtually everything in cell: active transport, biosynthesis, cell division, muscle construction. Beyond the cell, respiration is constant cornerstone of ecosystem. The carbon dioxide release feedback into the global carbon cycle. On the practical level, understanding respiration explains phenomena like muscle fatigue during exercise and the need to control respiration rates in feverish storm. So, conclusion and outlook. In summary, cellular respiration is a masterpiece of biological engineering. It strategically breaks down glucose through glycolysis, the citric acid cycle, and finally uses the electron transport chain to create a proton gradient that drives ATP synthesis via chemiosmosis. The principle of chemiosmosis is a unifying concept in bioengineering. Future frontiers in mitochondrial disease and metabolic engineering are built on this foundational knowledge. The continuous hum of this molecule machinery is the sound of life itself. Thank you for your attention and see you next time.